Thank you. So, hello everyone. My name is Suraj Bansal. I am an undergraduate student at McMaster University, which is in Toronto, Canada, and I'm also a research student in the Dr. John Dick Lab at Princess Margaret Cancer Center University Health Network, where we study a lot of things like stem cells and leukemia from a variety of approaches, including bioinformatics and experimental approaches. And so today I'm going to be presenting ATLAS-AML, which is an automated bioinformatics pipeline I have developed for rapid characterization of genes and gene signatures in acute myeloid leukemia. So to begin, I want to provide a little bit of background on acute myeloid leukemia, or AML. So AML is a hematological cancer that is well characterized by its very poor survival rates and is well documented for its heterogeneity. And I think that this concept of heterogeneity in AML has been looked at from three primary perspectives over the past couple of decades. The first is looking at interpatient heterogeneity and how patients with different genotypes or cytogenetic alterations will respond differentially to chemotherapy and various treatments. The second perspective is intra-patient heterogeneity, and this is how within a patient's blast of cancer cells, there are actually various different types of cancer cells, and it is the leukemic stem cells that are the ones that eventually become resistant to chemotherapy and will survive chemotherapy to then regenerate the disease and give rise to relapse. And the third perspective of looking at heterogeneity actually comes from a Nature Medicine paper that came out from our lab recently, which really discovers how within that whole blast of cells, how the patients actually organize into a hierarchical like structure with stem cells at the apex. And we were able to find that there are some patients that have a cellular hierarchy that is more stem cell driven, and others that have cellular hierarchies that are more mature cell type driven. And it is actually the patients in AML where properties of all three of these different perspectives kind of coalesce that are the ones that become resistant to chemotherapy and will ultimately relapse. And I think that this really highlights the inadequacy of standard induction chemotherapy in curing somebody's AML and highlights the importance of more targeted approaches in the clinic. And so speaking of this kind of concept of drug development in the clinic, there's no doubt that single cell RNA sequencing, where we've been able to take mRNA transcripts of individual cells in a patient, has really transformed the way that we've begun to study hematopoiesis and acute myeloid leukemia. However, with the immense amount of volume of data that is generated from single cell RNA sequencing, all of the subsequent analysis often requires somebody with a lot of bioinformatics or computational expertise to perform downstream analysis. And in requiring both this wet lab scientist and a computational scientist, it breeds a bit of a discrepancy in the landscape of drug development in AML, where we have a lot of wet lab scientists that are performing these time and resource intensive experiments that are often not informed by predictive analysis from the computational people, and vice versa, where we have a lot of good predictions that are made from bioinformatics researchers that sometimes remain unvalidated without the functional buy-in from those wet lab scientists. And so, hoping to kind of bridge these groups of scientists together, we have started to develop Atlas AML, which has taken between 20 to 25 in-house and publicly available bulk and single cell transcriptomic data sets and streamlined the analysis of genes and gene signatures across a multitude of phenotypes that go all the way from cell type associations at the single cell level to more clinical level analysis like prediction of patient survival and drug response. And so to paint some sort of broad strokes of what Atlas AML and streamlining all of these data sets hopes to accomplish is we want to help scientists that are researching a drug answer three fundamental questions. The first is that within that whole hierarchy of acute myeloid leukemia cells, specifically which cells is that drug targeting? And this is an important analysis to show because as we explained earlier, it's the leukemia stem cells that are the primary drivers of the disease. The second thing that we're able to help scientists figure out is within all of the patients, specifically which patients would benefit the most from this drug. Um, and we do this by essentially doing gene associations with all of the different mutational and cytogenetic profiles in acute myeloid leukemia. And this can help scientists not only figure out if their drug is viable in the disease, but also which patients to prioritize in clinical trials. And finally, the last major question that ATLAS-AML is intended to answer is just by doing univariate and multivariate associations with survival analysis to figure out whether this drug will actually confer better patient prognosis or not. And so now that I've kind of painted the broad strokes of what ATLAS-AML hopes to accomplish, I want to take you guys through each kind of step of the data and really what this pipeline can do. And so looking at this gene as an example here, DNMT3B is a gene that has been well documented to be a hallmark of acute myeloid leukemia at the transcriptomic level. And in all of the slides that I'm about to show, I'm going to show you exactly the type of analysis that ATLAS-AML can output for an individual gene that we query. Now, the first thing that we are able to look at in ATLAS-AML is its expression across the normal hematopoietic hierarchy. And this is cord blood that we process in-house and sort into various different fractions. And so in that way, we're able to look at the expression of our gene of interest across the normal hematopoietic hierarchy. And so in our example here, DNMT3B, we can quite strikingly see that it seems to be a marker of more hematopoietic stem and progenitor-like cells. The next thing that ATLAS-AML can do is it takes this normal hierarchy of cells and it isolates the stem cell and progenitor compartment, and it compares the expression of our gene in those normal stem cell and progenitors to an equivalent data set of leukemic stem cell and progenitors. And in this analysis, we're able to do direct comparison of gene expression in the normal versus the leukemic stem cells. And this is a really important analysis for scientists doing drug development because it helps them to isolate drugs that have higher expression in the leukemic system. Now, after looking at the expression of our drug in the normal hierarchy and the reference to leukemia, we can do kind of a similar approach, but in our single cell AML data. And so this cohort is 12 diagnostic AMLs from the Broad Institute that were subjected to single cell RNA sequencing, and then we refine the classification of those cells all the way from those that are more stem cell-like in the leukemic hierarchy to those that are more mature-like. And in this way, we're able to plot the expression of our gene of interest, DNMT3B, across the whole hierarchy, just as we've done before. And we're able to see that it's quite a clear marker of the more stem and progenitor-like leukemia cells. Similarly, as we showed before, where patient hierarchies can be classified into those that are primitive, which are more stem cell driven, and those that are mature, which are more mature stem cell like driven, we can look at the gene associations across these different types of hierarchies. And consistent with what we've been seeing so far, we see that DNMT3B, or gene of interest here, is overexpressed in those with a primitive or stem cell driven hierarchy. Next, to really hammer in on that aspect of the leukemia stem cells, we have data in-house that's 110 acute myeloid leukemia fractions that we reevaluated for functional leukemia stem cell activity via xenotransplantation. And so we're able to look at the expression across the leukemia stem cells that engraft and those samples that don't. And we're able to see quite a striking association with the engrafting leukemia stem cells in our example here. And we can go one step further looking at this by looking at its expression across the various different fractions and also seeing how it correlates with the actual frequency of leukemia stem cells. Next in ATLAS-AML, we can look at the expression at diagnosis versus relapse. And so these are 5 publicly available data sets that we took and compiled. And in all 5 of these data sets, the scientists performed bulk RNA sequencing at diagnosis, and then again for the patients that relapse. And so in this way, we compiled 68 paired samples at diagnosis and relapse, and we're able to look at the expression in these conditions as well. And again, consistent with what we've been seeing so far in our example gene, DNMT3B seems to be overexpressed in the transition to relapse. So everything I've shown up until now has been kind of to answer that first kind of arm of what I showed you earlier in the three-factor model. And that was all to kind of help scientists first figure out whether their drug is therapeutically viable. And so the second question, if you remember, was to figure out specifically which patients would benefit from that drug and who should be prioritized in the clinical trials. And so combining bulk transcriptome data from 3 clinical cohorts that are publicly available, we took all of the different mutational profiles, or all of the different mutation combinations, as you can see on the left here, and we looked at the expression of our gene across all of those mutational profiles. And this is a really important analysis because it can help provide a clinically and functionally relevant roadmap of which patients would benefit from a therapy against our gene the most. And this becomes really important because if we look at this graph, we see that the patient genotypes where DNMT3B is expressed the highest seem to be a lot of patients that have like a DNMT3A or FLT3-ITD mutation. Whereas patients at the top here that seem to have combinations including an NRAS mutation actually don't seem to express DNMT3B too much. And so in that way, we're able to kind of figure out which groups of patients might benefit most from a drug that targets DNMT3B. On a similar line of thinking, we're able to do the same thing for the different cytogenetic alterations that a patient can have, and also off of the primitive to mature blast morphology, which are all kind of clinical sorts of classifying patients that are used in the clinic right now. The next thing that we're able to look at is the overall connections with patient survival. And so these are 4 separate survival cohorts that we can look at overall patient survival in. The left 3 here are all adult cohorts, and then we've also included 1 pediatric acute myeloid leukemia cohort just because of how biologically different pediatric and adult AML are. And over here, actually, we see quite an interesting example because for our gene of interest, DNMT3B, in the leftmost cohort, it doesn't seem to be statistically significant. In the second cohort here, it seems to be very mildly significant. And in this third one, this final adult cohort here, it seems to be extremely significant. And so this can become a little bit hard to interpret whether it's overall significant in survival or not. And so taking this one step further, one of the final things that ATLAS-AML can do is it actually takes the hazard ratios from all 3 of those cohorts and using a package called METAP performs a meta-analysis of sorts that takes into account the hazard ratios and also the variance to generate a combined hazard ratio. And so the first thing that it can do is just univariately evaluate the hazard ratio, in which we can see that it's still significant and a predictor of survival. But taking this one step even further, we do it in a multivariate survival analysis as well. And this is where we can take into account clinical variables like age and white blood cell count and various mutations that are already associated with poor survival. And seeing that our gene here remains an independent prognostic factor from when we take into account all of these clinical variables lends to its significance, and that it might be capturing something that these other variables are not. And so to kind of summarize all of the type of analysis that our pipeline was able to give us on just that one gene that we were interested in, we were able to quickly determine that DNMT3B, our gene of interest, was correlated with leukemia stem cells in patients that had a primitive AML hierarchy. We were able to associate it with very clear phenotypes at the genomic, the cytogenetic, and the morphological level. And we were also able to see how it facilitates or is predictive of overall patient survival. And this is all analysis that I showed kind of on the previous slide, and that we can quickly regenerate for any gene that a researcher is interested in the context of acute myeloid leukemia. And so the next step in terms of where we are in this project right now is taking all of that analysis that I've shown today and streamlining it into an easy to use, publicly available web application where any scientist that's researching a gene or a drug in AML can just input that gene into the pipeline and basically get all of the analysis I showed today with just the click of a few buttons. And so that concludes my presentation. I'd like to acknowledge not only the organizing committee of this great conference today, but also all of the members of our lab that helped in putting this together, as well as I'd like to acknowledge the 7 labs that are currently using ATLAS-AML in collaboration with right now. And yeah, thank you very much. I'm open to any questions.
Thank you very much, Suraj. We do have room for one small question, if any. Yep, there's one there.
Hi. So you've clearly put a lot of work in, or your group put a lot of work into integrating and processing these different data sets for AML, which is a really valuable resource. So are you planning to make those data sets available for the community, which of course then accelerates discovery for the patients?
Okay, the question is if you are planning to make it publicly available for the scientific community, right? Yeah.
Sure question.
Thank you.
Pardon, could you repeat the question for me?
You're planning to make this data set publicly available for the scientific community?
Oh, yes, of course. So something that we're planning on doing in the near future is we're actually going to have a web application up and running where you can access all of the data I presented today, and also plug in a gene or a gene signature and get all of those plots kind of readily made as well.
They are asking for download and use it.
Pardon?
They're gonna be available for download and use it like locally.
Oh, yes. So you'll be able to download all the plots from the portal.